This protocol demonstrates the utility of the stem cell-derived intestinal organoid system for modelling epithelial invasion by enteric pathogens, and modification of this process using cytokines. Microinjection allows the pathogen of interest to be delivered directly into the luminal cavity of the organoid, replicating more closely the process of infection in vivo.
This method could be applied to the study of different enteric pathogens or alternative cytokines of interest. I would suggest completing a trial run of the procedure with phenol red only, in order to master the microinjection technique prior to using pathogens.
On day 2, begin the differentiation by changing the media to 10 ml of stem cell culture medium, supplemented with growth factors. On day 6, change the media to 10 ml of RPMI B27 media, supplemented with 6 micromolar CHIR 99021 plus 3 micromolar retinoic acid to begin patterning the posterior endoderm to the hindgut. On differentiation day 10, embed the hindgut in the basement membrane matrix. First, remove the media from the hindgut plate and wash the plate with calcium-magnesium-free DPBS once. Add 5 ml of collagenase solution to the plate and incubate at 37 degrees Celsius for 5 minutes. Next, inactivate the collagenase by adding 5 ml of the organoid-based growth media to the plate. The hindgut cells should be floating at this point. Collect the hindgut suspension in a 15 ml conical tube. Then centrifuge at 240 × G for 1 minute.